Hi there. Welcome back to our podcast. Today we're talking about chapter 11. This is the Central Somatosensory System. This all comes from our textbook, Neuroscience Fundamentals for Rehabilitation by Lundy-Ekman. So, in the last chapter, in the last podcast, we focused on somatosensory receptors in the skin and musculoskeletal system, and the first order neurons that bring that information into the central nervous system. This chapter now is about what happens after that, how the central nervous system processes somatosensory input, and why it matters for real movement and real function. So, somatosensation is not extra information. It's a major part of how movement works, even though it's sensory and not motor. You can have normal motor power and still be unable to do basic tasks like writing, holding a cup, or buttoning a shirt if you don't have somatosensory input. And this is because you lose that automatic, ongoing correction ability that keeps movement accurate and efficient based on sensory feedback. Somatosensory information also protects us against injury. When someone can't feel very well, they're more likely to develop pressure-related skin lesions, burns, and even joint damage because the threat doesn't register early enough to trigger an automatic response. For example, in something like congenital insensitivity to pain, people may repeatedly injure themselves, or develop fractures and deformities, and they can even die young. This is a unique circumstance. It doesn't happen very often, but it shows us what happens when we don't have normal somatosensation, especially in regards to our ability to perceive pain. So the key distinction across pathways now is how accurately the system can convey stimulus location and detail. And these pathways are the pathways that somatosensation travels peripherally to our central nervous system. High accuracy transmission in these pathways gives you precise localization, while low accuracy transmission is less specific. This is an example of this would be aching pain. That's a classic example of information that isn't well localized. So when we describe pathways, we usually count projection neurons. These are the long axon neurons that connect distant regions. The term tract is just referring to a bundle of axons with a shared origin and termination. And somatosensory pathways are often named based on where the second order neuron's tract starts and ends. From there, the chapter then organizes for us somatosensory transmission into three pathway types. We have conscious relay pathways, divergent pathways, and non-conscious relay pathways. Conscious relay pathways transmit information to the cerebral cortex with high accuracy. They support fine discrimination, but not everything that reaches the cortex is actually consciously perceived. Our attention has to be directed to the sensory input in order for it to enter our conscious awareness. Divergent pathways send information to multiple areas of the brainstem and the cerebrum, and they use variable numbers of neurons, and they support both conscious and unconscious responses. Aching pain is one example of sensation commonly transmitted this way. Now, non-conscious relay pathways carry proprioceptive and movement-related input to the cerebellum and are essential for automatic postural and movement adjustments. All four somatosensory modalities can reach conscious awareness, and the spinal cord sends conscious information to the brain using two main routes, the dorsal columns and the anterolateral tracts. So our conscious relay pathways have two main routes upward, and they're through the dorsal columns of the spinal cord and the anterolateral tracts in the spinal cord. Let's start with light touch and conscious proprioception. This is travels in something we call the dorsal column medial lemniscus system. Light touch, which is also called discriminative touch, includes localization of touch, vibration, and the ability to distinguish two closely spaced points. Conscious proprioception is our awareness of movement and the relative position of our body parts in space. A key functional outcome that travels in these tracks is something called stereognosis, which is our ability to identify an object using touch and proprioceptive input without vision. An example is the ability to recognize the shape of a key in your hand. Maybe when you're going to unlock a door, you pick up your keychain, and without having to look at it, you're able to manipulate the key that needs to go in the lock into your hand. This relies heavily on our ability to use stereognosis effectively. This pathway, this light touch conscious proprioception pathway, is classically described as a 3-neuron chain. The primary neuron carries information from receptors in the spinal cord through the dorsal root, and it ascends ipsilaterally. This is important. It ascends ipsilaterally in the dorsal columns to the medulla. Fibers from the lower limb and lower trunk in this tract travel through what we call the fasciculus gracilis, and they synapse in the nucleus gracilis. Fibers now from the upper trunk and upper limb and neck travel in the fasciculus cuneatus and synapse in the nucleus cuneatus. All of these terms are part of that dorsal column medial lemniscus tract. We're just changing what we call the individual like bundles of axons that they travel in based on where they're originating from. So once again, fibers from the lower limb and lower trunk travel in the fasciculus gracilis. This is in our spinal cord, and they synapse then in the medulla in what we call the nucleus gracilis. Opposed to this, we have the upper trunk, the upper limbs, and the neck. Information from those body parts travel in the fasciculus cuneatus and synapse in the medulla in the nucleus cuneatus. Again, this is all part of that DCML, dorsal column medial lemniscus. The secondary neuron begins now in the medulla in the nucleus gracilis or cuneatus based on where the information originated. It then decussates, which is a really important term here. Decussates means cross. So now it crosses to the contralateral side. Remember, this information originally was traveling in the spinal cord on the ipsilateral side. So it travels in the spinal cord on the ipsilateral side, synapses now with the secondary neuron in the medulla with either the nucleus gracilis or cuneatus. It then decussates, so it crosses over now in the medulla and it ascends to the thalamus now as a contralateral medial lemniscus. From there, it synapses in something called the ventral posterolateral nucleus, or the VPL. The VPL is in the thalamus. Now we are on our third neuron, which projects from the VPL to the cerebral cortex through the thalamocortical radiations in the internal capsule. Once the information reaches the cortex, the primary somatosensory cortex receives this somatotopically organized input. So this information that goes from the thalamus to the cortex goes to a different location of that primary somatosensory cortex based on which part of the body it comes from. So our information goes to this somatotopically organized cortex. And in this primary somatosensory cortex, we have a discrimination of size, texture, and shape being initially perceived here. The secondary somatosensory cortex helps us to analyze input from the primary sensory cortex and the thalamus, and it supports stereognosis and memory for tactile and spatial experience. This is where the homunculus concept comes in. And if you look at our cover photo for this project, you'll see a funny little guy that we can call Mr. Homunculus. He represents this homunculus concept. So what is the homunculus? Well, it's a mapped representation of the body surface in the cortex, in the somatosensory cortex, that shows us where different parts of the body are represented in that cortex. And our mapping of the homunculus is actually derived from recordings in awake individuals during surgery, showing how much cortical space different body regions perceive. You'll notice that the hands and the mouth are take up a much larger representation of that somatosensory cortex than other parts of your body. Now we're going to move on from the DCML, dorsal column medial lemniscus, to what's called the anterolateral system, which carries information about fast nociception, temperature, and crude touch. And this travels through something called the spinothalamic pathway. For the anterolateral system, the chapter emphasizes that nociception, temperature, and crude touch signals can travel through conscious relay and divergent pathways. And it also draws a clear contrast now between fast versus slow nociception. So the spinothalamic pathway is the classic conscious relay route for discriminative fast nociception, as well as temperature and crude touch. Just like the DCML, this is a 3-neuron pathway. The first order neuron enters the dorsal horn, and the second order neuron crosses the midline and projects from the spinal cord to the thalamus. So we have this spinothalamic tract now entering into the spinal cord through the dorsal horn. From here, it immediately synapses with the second order neuron and crosses or decussates. So while the DCML did not cross until the medulla, the spinothalamic tract immediately crosses as soon as it enters that spinal cord. Now we're here with the second order neuron, which projects from the spinal cord up to the contralateral thalamus. And the third order neuron now projects from the thalamus to the cerebral cortex. More specifically, the first order neuron cell body is in the dorsal root ganglion, and fast pain typically travels on an A delta fiber. Temperature detection involves specialized free nerve endings in small myelinated and unmyelinated neurons, and crude touch carries non-light touch information. The second order neuron cell bodies are in the dorsal horn, and their axons form the spinothalamic tract and decussate, remember that means cross, in the anterior commissure of the spinal cord. Many of these axons end in the VPL, and third order neurons project to primary and the secondary somatosensory cortices. Now, functionally, localizing noxious stimuli depends heavily on the fast nociceptive system, also called the lateral system. And lesions affecting the VPL can actually interrupt the pathway then to the cortex. Crude touch travels alongside fast pain in the spinothalamic pathway, still using the 3-neuron pattern, but it conveys non-discriminative tactile information, often via C fibers, and it contributes to the awareness of contact without precise localization. It also projects to the insula. A helpful comparison is that both the dorsal column medial lemniscus and the spinothalamic pathways are anatomically similar in the 3-neuron VPL to cortex sense, but they differ in what is traveling where and where they cross. The dorsal columns contain primary neuron axons that ascend ipsilaterally and decussate in the medulla, while the spinothalamic tract contains second order axons that decussate in the spinal cord and ascend contralaterally. Okay, moving on now to slow nociception. Slow nociception is framed as a medial nociception system that uses divergent pathways running in the anterolateral columns alongside the spinothalamic tract. Slow nociception contributes to affective and motivational dimensions of pain, as well as withdrawal, arousal, and autonomic responses, and many projection neurons synapse in more medial brain locations. The slow system here is different from the fast system because it uses separate neurons, synapses in different brain areas, and it often involves multiple pathways with variable numbers of projection neurons rather than a simple 3-neuron chain. It also isn't somatotopically organized, so it's harder to precisely localize. Our textbook breaks the ascending projection components into three parallel tracks. We have the spinomesencephalic tract. There's a lot of big words in this one, guys. All right, so we got three parallel tracks: spinomesencephalic, spinal reticular, and spinolimbic. Only information traveling in the spinolimbic tract is typically perceived as aching or localized pain. Oh my goodness. Let's try again. These spinomesencephalic projections target the superior colliculus, which helps us to orient our eyes and head toward a painful source, and also to something called the periaqueductal gray, which is part of the descending pain control system. Spinal reticular projections synapse in the reticular formation, and this influences arousal, attention, and sleep-wake regulation, with downstream projections toward the intralaminar thalamic nuclei to areas including the anterior cingulate cortex and the insula, and then onward to prefrontal and parietal cortices. Spinal limbic projections transmit slow pain information to medial and intralaminar thalamic nuclei and to the ventral striatum, supporting automatic movement plus autonomic and emotional responses to noxious stimuli. So let's talk about those again before we move on. Spinomesencephalic projections go from the spine to the midbrain, where we have the superior colliculus. That tract is important in helping us immediately orient our eyes and our head toward the source of a noxious stimulus. The spinal reticular projections go from the spine to the reticular formation, and that helps us alert and draw our attention immediately towards that pain. Then we have the spinal limbic projections, which go from the spine to our limbic system, which is involved with our emotion system. So this is why we have a painful stimulus affecting our emotions. Okay. So for the dorsal column medial lemniscus, if we have a lesion to that system, the site of loss is going to depend on whether the lesion is below or above the decussation in the caudal medulla. Below the decussation, you see ipsilateral loss of discriminative touch and conscious proprioception. For spinothalamic tract lesions, because decussation occurs in the spinal cord, lesions cause contralateral loss of pain sensation below the lesion. Pain is often linked to tissue damage or threat of damage, but pain can also occur without tissue damage. Nociceptor activity alone isn't enough. Nociceptive signals must also be processed by the brain to become pain, and pain includes both perception and emotional response. Deep pain from muscles and joints is different from superficial pain because it often appears after tissue damage and can function to encourage rest. Clinically, it can alter movement patterns. For example, pain with weight bearing after a lower extremity injury often produces a modified gait with a shortened stance phase affected on one side. This is often described as an antalgic gait, meaning a gait that adjusts to avoid pain. Referred pain is described as pain perceived at a location different from the true origin, often referred from the viscera to the skin. The mechanism emphasized here is convergence. Nociceptive information from the skin and internal organs converges on the same second order neurons in the spinal cord or the same third order neurons in the thalamus. The pain matrix is presented as a set of brain structures that process and regulate pain information and can generate pain perception even without nociceptive input. It includes parts of the brainstem, the amygdala, hypothalamus, thalamus, and cerebral cortex. Understanding pain requires considering three components. We have the discriminative component, which is involved with localization, timing, and intensity of the pain. We have the motivational affective component, and this is the effect of the pain on emotion and behavior, like arousal and avoidance. We also have the cognitive evaluative component, and this is the meaning that the person assigns to that pain. I think the moral of this story to take away from this little section of our podcast is that pain is perceived differently by everyone. So when you have a patient or a client and you're asking them about their pain, regardless of whether or not you think you understand how much pain they may be experiencing or what injury they have, how painful that injury typically is. It's important that you understand pain is not just about the pure tissue damage. Pain is dependent on that individual's unique perception. It's a complex system. Okay, moving on to how pain is controlled now. So, our slides, our lecture slides from this chapter, use a simple example. When you hit your thumb, you instinctively apply pressure. You bonk your thumb or you hit it with a hammer. That's kind of the classic, like Homer Simpson example. After that happens, you instinctively want to apply pressure to it. This stems from something called the gate theory of pain. And that was an early explanation of how pressure and external stimuli can actually inhibit pain transmission. The parts of the original gate theory of pain were incorrect, but this theory was important because it drove later work on pain control mechanisms and clinical tools like the TENS unit. Pain processing can shift between normal, suppressed, and sensitized states. In the suppressed state, touch, pressure, and vibration may transmit normally, while the nociceptive impulses are actually inhibited. In the sensitized state, opposed to that, we have changes in neurotransmitters and receptors that can produce painful responses to signals that wouldn't normally be painful. The sensitized state is tied to something called central sensitization. Central nervous system neurons actually show increased responses to nociceptive and non-nociceptive stimuli during this sensitized state. So after acute tissue injury, peripheral sensitization can trigger central sensitization, and this then strengthens synapses and amplifies incoming nociceptive messages to make them more painful than they typically would be. So there's two important terms to discuss, and these are antinociception and pronociception when we talk about pain. Antinociception is defined as suppression of pain in response to stimulation that would normally be painful. Transmission of nociceptive information can be inhibited by pain matrix activity, and the cerebral cortex can activate pain suppressing mechanisms via the descending cortico-periaqueductal tracts. Remember, I mentioned the periaqueductal gray and its involvement in our antinociception system. That's where we're coming from here. So it's a descending cortico-periaqueductal tract. Intrinsic antinociception involves descending systems from brainstem regions, including the rostral ventromedial medulla, with raphispinal tracts releasing serotonin in the dorsal horn, and the periaqueductal gray, which activates the RVM. Another brain region involved with antinociception is the locus coeruleus, with ceruleospinal influence that inhibits spinothalamic activity by suppressing nociceptive neurotransmitter release. We also have endogenous opioids, such as enkephalins, dynorphin, and beta endorphin, which activate antinociceptive mechanisms. And opioid receptors are found in areas including the thalamus, the periaqueductal gray, of course, the RVM, and the dorsal horn. So stress-induced antinociception is also important. Stress can activate all of these systems. It can trigger the raphispinal activation, which remember that's involved with our intrinsic antinociception system. And it can drive hormonal release, including beta endorphins, with effects that can last for hours. So there are 5 key sites or levels of antinociception. We have it happening in the periphery, in the dorsal horn, in the fast neuronal descending systems, in the hormonal systems, and at the amygdala and cortical level. On the other side, as opposed to antinociception, we have pronociception, which is the biologic amplification of nociception. Pain transmission can be intensified at multiple levels. Peripherally, we can have edema and endogenous chemicals that can sensitize free nerve endings, making them more responsive to pain. Pronociception can also occur when someone is anxious or depressed, and that pronociceptive pain matrix activity can contribute to pain perception even in the absence of nociceptive input. That's why it's important to take pain seriously when someone reports it, because their perception of pain is unique to them. Okay, so to summarize all of this up, discriminative touch and conscious proprioception travel via the 3-neuron DCML pathway, dorsal column, medial lemniscus. Fast nociception, temperature, and crude touch travel through a 3-neuron spinothalamic pathway. The lateral or fast pain system supports localization of tissue threat and damage, while the medial slow pain system emphasizes affective and cognitive dimensions and uses divergent pathways. Pain can also be amplified or suppressed depending on the central processing state. All right, that's a big one, but honestly, it's one of my favorite chapters in this book. Okay, talk to you next time. Bye-bye.